Hello, everyone. Hello, everyone, dear respected colleagues. I see in the participant list from so many latitudes, from America to the far Asia. So that points out that the interest about the topic of this webinar, that is the fourth webinar in a series of we have programmed at the INSAS network. The topic is of high interest. And I'm sure that all of we will learn from the points that will be presented by so distinguished panelists that we have today. So the topic, as it was announced, it is the crop nutrition under salt-affected conditions. And the speakers of today will discuss different aspects relative to this subject, including the following points that are nutritional problem of salt-affected soils, deficiencies and toxicities, nutrient behaviors in salt-affected soils, crop nutrition management in salt-affected soils, nutrient and crop-wise management practices, and they will present as well some case studies on crop nutrition management in salt-affected soils. This webinar will be enriched by the participation of the attendants with questions and comments that you can type in the chat window, and they will be answered in due time by the speakers. And now allow me to introduce them, the bios of the speakers, and I will do that in an alphabetical order because all of them have a wide curriculum, and so they are same level of excellency. First, allow me to introduce Dr. Nadia Vacigina. She is a plant ecophysiologist whose work focuses on understanding how salt-allowing halophytes thrive in saline environments. After a PhD at the University of Western Australia, she has had several postdoctoral positions in Australia and Italy in plant stress physiology, including the European Union-funded Global Marie Curie Fellowship, focusing on the halophytic crop quinoa. She's currently an assistant professor in plant physiology at the University of Florence and vice chair of the Coast Action Sustain, an international network of scientists working on saline ecosystems and how to sustainably manage them. Dr. Nadia Vacigina, you are welcome. Next, I would like to introduce to Dr. Muhammad Saqib, the promoter of this webinar. We are very, very grateful to you to organize this webinar. He is a professor at the Institute of Soil and Environmental Sciences, University of Agriculture, Faisalabad, Pakistan. He has been working on saline agriculture for sustainable use of salt-affected soils for the last more than 20 years. He is a fellow of Alexander von Humboldt Foundation, Germany, and fellow of Japan Society for the Promotion of Science. He has worked as Justus Liebig University, Gießen, Germany, University of Tokyo, Japan, and University of York, United Kingdom. Dr. Muhammad Saqib, you are very welcome. Professor Steven Schubert is Emeritus Professor of Plant Nutrition, still very active, not only in plant nutrition in general, but particularly in the field of soil salinity and salt resistant of crop plants. He was professor of plant nutrition at Justus Liebig University, Gießen, Germany, and head of the Institute of Plant Nutrition from 1997 to 2022. He has published 2 standard textbooks of biochemistry and plant nutrition in German. He has published 140 peer-reviewed articles and over 100 other contributions. Together with his colleague at University at Hamilton, Canada, Professor Dr. Mansour Kadir, he's working on a textbook on soil salinity and salt resistance of crop plants. He's an associate editor of the Journal of Plant Nutrition and Soil Science. Professor Sven Schubert, you are very welcome. Now it's the turn to introduce to Sivesvar Pratap Singh, a soil scientist whose work focuses on understanding management of on salt-affected soil, nutrient management, and natural farming. After getting a PhD at the G.B. Pant University of Agriculture and Technology, Pantnagar, India, he has had attended several meetings regarding salt-affected soils and also involved in teaching to undergraduate, postgraduate, and doctoral students. He is currently an associate professor in soil science at Dr. Rajendra Prasad Central Agricultural University, Pusa, India, and also member of the International Network of Salt-Affected Soil, INSAS. Dr. Pratap Singh, you are welcome. And at last but not least, allow me to introduce to those that are new today to the INSAS meetings, to our coordinator at FAO headquarters, Dr. Maria Kunioskova, who will take over the highlights of the meeting of today. So after this presentation, I would like to give the floor to the Professor Muhammad Saqib that will develop the topic nutritional imbalances in salt-affected soils. So, Professor Saqib, the floor is yours.
Okay, thank you very much, Professor Battle Sales, for the kind introduction. And I'm also very thankful to FAO as well as International Network of Salt-Affected Soils for giving me this opportunity to actually coordinate this seminar and to present in this seminar. And I would like to make my presentation on the nutritional imbalances in salt-affected soils. So in this talk, I will cover in general that what are the salt-affected soils and what are the different nutritional problems. I will try to cover briefly most of the nutrients which have certain interactions, like we have sodium toxicity in salt-affected soils, chloride toxicity in salt-affected soils, then nitrogen, phosphorus, potassium, calcium, magnesium, and different micronutrients. And at the end, I will present a few points with respect to the nitrogen nutrient management in salt-affected soils. As we know, the salt-affected soils are generally classified into saline soils, sodic soils, and saline sodic soils. Saline soils have high soluble salts. Saline sodic soils have high exchangeable sodium and high pH. And saline sodic soils, they have high exchangeable sodium and high pH. And saline sodic soils, they have high soluble salts as well as high exchangeable sodium and high pH. Now, the aim to show their characteristics here is that these characteristics of these salt-affected soils, they affect the nutrient behavior in the salt-affected soils. The high concentration of the salts, the high concentration of sodium, the exchangeable sodium, high pH. So all these characteristics, they have different effect on the nutrients, nutrient availability in the salt-affected soils. In general, if we look at the literature and our own experiences, they show that salinity-nutrient interactions, they are very complex. In certain cases, we see increase in the concentration of different nutrients. In some other cases, there is decrease. And even some cases, there is no effect on the concentration of different nutrients. Now, if we try to explore the causes or the factors on which these behaviors they depend, we come across few points that the nutrient behavior that depends on the composition of the salts, whether there are sodium salts, chloride salts, calcium salts, sulfate salts. So they all have their impact on the behavior of different nutrients. Then the level of the salinity, whether it is slightly saline soil, moderately or highly saline soil. So that level, that actually shows that how much will be the extent of the ionic competition in that particular soil. Then what is the nutrient concentration in the soil? For example, if we take example of nitrogen or phosphorus or potassium, whether they are deficient already in the soil, they are present in good quantity. So that also affects their ultimate availability to the plants. Then plant factors, including the species, the genotypes, transport of ions in the plant and partitioning of ions in the plant, they play important role in plant nutrition under sulfur conditions. And last but not least, the climatic conditions, including the temperature, including the relative humidity, precipitation. So all these things, they affect the nutrient behavior inside the salt factor size and it's available for the plants. To start with, I would like to discuss sodium toxicity in the salt factor size because this is the most important ion which is present in high concentrations in the salt factor size and it affects the availability of the other ions. So in our experiments on wheat, when it was grown not only under saline conditions, but under saline and waterlogged conditions, there was additional stress of compaction as well as waterlogging. So we found that sodium concentration that is increased in plant under salt-affected conditions. And when there is additional stress of waterlogging, there is even more significantly higher increase under these conditions. Similarly, and when we compared saline conditions and saline soda conditions, so in both the conditions, the concentration of sodium inside the plant that was significantly increased. Then in wheat, we compared different genotypes of wheat. So there was significant difference with respect to their sodium accumulation, as well as their capacity to translocate sodium from root to the shoots. So plant genotype or species, they have significant impact on the sodium uptake inside the plant under salt-affected conditions. Then it's well known now that silicon, it has positive impact when it is applied under different stress conditions. So as we found in wheat, the application of silicon that improves the plant performance under these conditions due to a decrease in the sodium concentration inside the plant. It not only decreases the sodium concentration, it also decreases the sodium distribution or sodium translocation from root towards the shoot. And we also found that this decreased effect of sodium was due to its binding with the cell wall when there was additional application of silicon under salt-affected conditions. Then the second most important comes the chloride. So chloride toxicity is also very widespread. And there is again difference with respect to plant species and plant genotypes. And in wheat, we saw that there is increase in chloride concentration under saline conditions. And when there is an additional stress of waterlogging, the concentration is significantly further increased inside the plant. And where we compare salinity and sodicity, so there was a decrease, there was less increase in chloride inside the plant under saline soda conditions, whereas there was high increase under salinity alone. In potato, it has been reported that due to chloride salinity, there is a significant increase in chloride concentration inside the plant when it is grown under sodium chloride salinity. If we look at the nitrogen interactions in sulfur sites, so it seems that nitrogen nutrition is compromised in sulfur sites, and that is because high concentration of chloride, it competes and reduces nitrate uptake. And high concentration of sodium, it competes and reduces ammonium uptake under sulfur conditions. And it is not only a competition of chloride and nitrate and sodium and ammonium. There is also decrease in the symbiotic nitrogen fixation, which is an important source of nitrogen under all the conditions, whether sulfur-affected or normal. So symbiotic nitrogen fixation is reduced in sulfur-affected soils because of reduced nodulation, rhizobium number, and efficiency, and due to reduced supply of photosynthets to the nodules. In this study, there was a very clear indication that due to increased salinity, sodium chloride salinity, there is a decrease in the nitrate nitrogen inside the plant. This is a study on the maize plant. So nitrogen, particularly nitrate nitrogen, that is decreased, whereas there is an increase in the chloride concentration. If we look at phosphorus and potassium interactions in sulfur soils, phosphorus availability is either decreased or increased, or sometimes there was no effect. So different studies have reported different results. With respect to potassium availability and uptake, it has been reported that this is reduced under sulfur soils. And this is because of high sodium concentration and because of disruption in the cell membranes. And it has also been seen that when there is a high supply of calcium, that improves potassium uptake and it reduces leakage of potassium as a result of improved membrane integrity. Our results on wheat, they have clearly shown that potassium concentration that is decreased under different stresses, including salinity, waterlogging, compaction, and under combined stress, it is further significantly decreased. In a study by Professor Schubert, it has been modulated that how under sulfur conditions, particularly sodium, it can to some extent substitute for potassium. So they actually proposed this guidance that to what extent and how we can actually move toward the substitution of potassium by sodium to some extent under sulfur conditions for some plants. For phosphorus concentration, again, our own work on wheat as well as work of other scientists on rice, they have shown a decreased concentration of phosphorus in rice and in wheat under salt-affected conditions. If we look at calcium and magnesium interactions in salt-affected soils, calcium availability and uptake is decreased in salt-affected soils because of ion interactions. And addition of calcium under salt-affected conditions that improves membrane integrity and it reduces leakage of different ions and also reduces sodium toxicity. With respect to magnesium uptake and accumulation in plants, it is either reduced or not affected by salinity, as has been reported in the literature. However, calcium and magnesium, they compete, and magnesium deficiency because of high calcium that has been reported. In our studies on wheat, we compared the performance of different wheat genotypes under saline conditions and under low calcium conditions and saline and low calcium conditions to know the effect of calcium on salt tolerance as well as accumulation of sodium and calcium under these conditions. So we found that under saline conditions, there is decrease in the calcium concentration, calcium uptake by the plants, and there is increase. When there is a decrease in the calcium availability inside the plant, there is a further increase in the sodium uptake by the plants. So sodium accumulation inside the plant that decreases with a decrease in the calcium availability under salt-affected conditions. If we look at the micronutrient interactions in salt-affected soils, there is a very complex picture. In some studies, they have shown an increase under salt-affected condition for some micronutrients. In most of the studies, there has been a reduction in micronutrients. But again, it depends on the plant. It depends on the nutrient which we are considering. It depends on the concentration of the salt, the salinity level inside the soil or the substrate. That is very important with respect to these results. So some of the references in the literature and our own work that shows that salinity reduces manganese uptake in corn. It decreases zinc accumulation in corn. However, in rice, an increase in zinc, copper, and manganese has been reported under saline conditions. And the authors suggested that it is due to decreased pH under these conditions. Similarly, our own studies have shown that salinity decreased iron concentration in rice. And when there was low iron, so the salt tolerance of the plant that was also reduced. With respect to other micronutrients, an increase in molybdenum concentration due to salinity has been reported. A decrease in copper concentration in corn and an increase in copper concentration in tomato has been reported. Salinity and boron interactions, they are very famous and they have been found in wheat, maize, and sunflower in our own studies. A decrease in uptake of boron under high calcium and sulfate that has been reported in wheat. These are studies on rice. One is from our own lab. So in this case, when we exposed the plants to salinity under iron, normal iron and iron deficient conditions. So under deficient conditions, there was increase in the shoot sodium concentration as compared to the normal conditions where the normal iron concentration that was present in the substrate. Whereas there was a decrease under these conditions for the potassium. So potassium under iron deficient conditions, that was less in the plant, its concentration significantly less as compared to the saline conditions, whereas sufficient iron was present. Whereas another study, this is just contrasting on rice again. So they have reported an increase in these nutrient concentration of these nutrients, including zinc, copper, and manganese. Again, they compare two genotypes, and in both the genotypes, there was an increase in the concentration of these nutrients under saline conditions. This is again our own study where we actually added silicon under saline conditions, and we observed the behavior of maize under these conditions. And with respect to iron and zinc, when we applied silicon under saline conditions, so there was an increase in the concentration of zinc as well as iron under saline conditions by the addition of silicon. If we look at boron interactions in salt factor conditions, so we saw in wheat as well as in maize that these salinity and boron, when they come together, they have a more negative impact on the growth of wheat. And if we look at the ionic concentrations inside the plant, so higher sodium concentration was found in maize when there was uh, additional stress of boron. And in case of boron concentration, again, the trend was that when there was additional stress of salinity, that increased the boron concentration inside the shoot. So there was a synergetic effect of salinity and boron in wheat as well as in maize. Now, towards the end, I would like to talk about a few points on the nutrient management in salt pesticides. So as we saw that nutrient behaviors in salt pesticides, that is very complex, and that depends on many factors. So likewise, it is not easy to manage the plant nutrition under salt factor conditions. We need to actually follow all the general management practices, which we do for soil, water, as well as crop management. Then we use the soil amendments to reduce the salinity level, which we can call that the different reclamation practices and rehabilitation practices that will reduce the salt effect and that will improve the nutrition under these conditions. Then addition of organic matter in all its form, whether it is compost or it is farmyard manure or in any other form, green manuring. So that will certainly help in reclaiming the soil, rehabilitating the soil, as well as in improving the nutrition under these soil conditions. Then the use of salt-tolerant plant species and genotypes. That is also a key factor. So as we have seen that there is a significant interaction with respect to different nutrients for different genotypes and different plant species. So we need to actually use the salt-tolerant plants for salt factor sites. And of course, it's difficult for the farmers particularly to use additional fertilizers under these conditions. But as the nutrition is compromised, so we do need additional fertilizers in addition to the recommended doses of fertilizers, particularly for the macronutrients, including nitrogen, phosphorus, and potassium. So by these nutrient management and other management practices, we can have better nutrition under salt factor conditions. So with this, I would like to stop here and thank you very much.
Thank you very much, Dr. Akib, because of your interesting review on the interactions of ions under salt-affected conditions. And well, including as well waterlogged conditions that it is not always considered by reviewers, but it is interesting as well. And you got several questions that, on sake of the profit of time, will be addressed to you at the end of the meeting, together with the questions to the other speakers. So thank you very much. And now allow me. Next speaker is excuse me, Professor Sven Schubert, that will develop the topic plant under salt stress. Is energy a limiting factor? So, Professor Schubert, the floor is yours.
Can you hear the? Can you see the screen?
Yes, perfectly, and hear you well. Thank you.
Thank you very much for your introduction. And I should like to thank the organizers for inviting me to give this presentation. As you all know, there are a lot of factors that negatively impact on the growth and yield performance of plants under soil stress. And Professor Sakib has already shown many examples how we can combat these problems. In many cases, it's either impossible to improve the soil conditions because the resources are scarce, for example, water availability, or at least it's very costly. And therefore, back in 1941, Lyon was the first to suggest that we should breed for salt resistance of crop plants. Of course, when we want to breed for better performance under saline conditions, we have to know what are the most important traits that we have to breed for. And one of these traits was suggested to be energy efficiency. And I should like to cite this article by Rana Mans and her colleagues. She investigated how much energy is used when plants osmotically adjust to the saline conditions, and how much energy is used when they exclude ions or compartmentalize them in order to avoid toxicity. They showed that there is a lot of energy used in addition to the energy that the plants usually needs. But the question is energy really a limiting factor for plants grown under saline soils? This is an important question because salt resistance is a multigenic trait, and we have to consider whether it's really necessary to look for this trait. Therefore, it is first of all important to consider her two-phase model, which she published in 1993. And according to this two-phase model, we can distinguish two different phases. When we add sodium chloride to the root medium, we can see that there's a decrease in growth rate. And this decrease in growth rate is explained by phase 1, which is an osmotic reaction of the plants. However, we must stress there is no deficit in the plant. Although water availability in the soil is decreased under saline conditions, the plants can adapt and there is no water deficiency. Growth is further decreased, especially in sensitive genotypes, in phase 2. And this phase is related to the outcome of ion toxicity. So we must distinguish these two different phases, and I think it's very important when we want to understand physiologically how the plants react to salinity, we have to distinguish these phases. There's another aspect, and that is when we suddenly, in a shock treatment, add sodium chloride to the root medium, for example, in nutrient solution, then we have a very strong decrease in growth rate. But the plants are able to recover within several hours to a certain level where we enter then phase 1 or phase 2. Now, this strong reaction, according to a shock treatment, is not relevant to salt resistance. That's why I introduced a phase 0 in order to avoid investigations in this phase 0. Unfortunately, many publications in the literature address this phase 0 and discuss possibilities of salt resistance of the plants. Especially when molecular biological work is done, many researchers are investigating phase 0, and this phase 0 is not relevant to salt resistance. Now, keeping this in mind, we first investigated whether there is a decrease in ATP concentration when we add sodium chloride to the plants. You see, this is a rather short experiment, and what it shows is that even at very high concentrations of sodium chloride, there is no decrease in the ATP concentrations of the roots. And this is very important, and it shows that apparently ATP as a general energy currency is not limiting under saline conditions. I want to stress this by showing another experiment which shows you the effect of nutrient addition to the ATP concentration. NS stands for nutrient solution, whereas calcium stands for calcium sulfate. And what you can see is, as soon as we add nutrients, and this is mainly phosphate and nitrogen, you can see a strong increase in the ATP concentrations relative to the calcium sulfate solution. What is the reason? The reason is that the production of AMP, the synthesis of AMP, requires nitrogen and phosphorus, and that increases the AMP concentration. And because the plants have to keep the energy charge, they produce additional ATP. So this is similar to the situation when we add a saline medium. We had two different other treatments in this experiment, and SR stands for split root, where we added nutrients only to half of the root system, whereas in this split root calcium, it was again calcium sulfate, we added no nutrients. And you can see the same result as in this case. As soon as we add the nutrients, there is an increase in ATP concentration. How can this happen? It was related to an increase in respiration, but not in alternative respiration or relative abundance of transcripts of uncoupling proteins. So why can plants so efficiently regulate their ATP concentrations? Now, first of all, they can increase their respiration, as I've just shown. Then you should keep in mind that plants usually do not do photosynthesis at their maximum, but there is always a sort of reduced photosynthesis, which can be increased as soon as there is a demand. Then plants have the unique feature of an alternative respiration, and this plays a certain part usually under normal conditions. Now, as soon as there is a stress, they can reduce their alternative respiration, which is less efficient than the conventional respiration, and increase the conventional respiration, so becoming more efficient in ATP production. There's another mechanism that allows that an excess of ATP can be avoided by uncoupling proteins. Now, in the inner mitochondrial membrane, plants of mitochondria, plants have the possibility to express uncoupling proteins. These uncoupling proteins reduce the proton gradient without ATP production. So again, it's an inefficient use of the proton gradient in order to produce ATP. And again, as soon as there is an additional demand of ATP, the uncoupling proteins can be reduced. And finally, we should not forget that plants release a lot of root exudates into the rhizosphere, which can also be reduced so that they can save energy. So by these different mechanisms, plants are able to increase ATP concentrations. However, the question arises whether whether there are enough assimilates from photosynthesis to guarantee ATP production also in the long term. And therefore, we investigated source-sink relationships in vegetatively and generatively grown plants. And what I want to show first is an experiment with wheat, which is a C3 species, and maize, which is a C4 species, more efficient in photosynthesis. And what you can see is that in both cases, under saline conditions, this is the red column, you can see a strong increase in sucrose concentration. Now, sucrose is a very important assimilate because it serves as a long-distance transport metabolite. And this increase in sucrose concentration shows that apparently there is no source limitation. So photosynthesis was high enough to deliver enough assimilates. And this is not only true under high illumination conditions, but is also true when we reduce the light intensity. You can see almost the same result under the low light intensity situation. So when growing plants vegetatively, you can see that apparently there is no source limitation, but a sink limitation. The sink limitation occurs, for example, because the key enzyme acid invertase is reduced. When sucrose is delivered by the phloem, it enters the apoplast, and in order to establish sink activity, acid invertase must hydrolyze sucrose into the hexoses, fructose, and glucose, and they can then be accumulated in the sink cells. Now, a decrease in acid invertase would just decrease the sink activity, and this can be actually the case under certain conditions, which was shown here in this experiment. You see two different maize cultivars, and the blue columns again show the control conditions, and the red columns show the salt stress. And what you can see here is the acid invertase activity in developing kernels of various maize hybrids. So we are in the generative phase during flowering, and what you can see is a strong decrease of the acid invertase in both cultivars. We did another experiment, and in this experiment we also investigated two cultivars of maize, Pioneer 3906 and Fabregas from KWS in Germany. And you can see that salinity in this case decreased the grain yield by about 1/3. And this was the case in both cultivars. However, kernel number explained this grain yield in this cultivar Pioneer 3906, but only partly in Fabregas. So apparently, the time during flowering is a very sensitive stage, and during this stage, a lot of grain yield can be lost, for example, by salinity, due to salinity in this cultivar. Less in this cultivar, whereas in this cultivar there is some decrease in kernel weight, which was not the case in the Pioneer 3906 cultivar. Now, showing the sugar concentrations, you can see that, as I've shown before, in the vegetative phase, there is no source limitation. There is enough photosynthesis because the sucrose concentrations are always higher under saline conditions, which means that under saline conditions, the consumption of assimilates was less affected, was more affected than the production of the assimilates. Surprisingly, we found that this was also the case for the hexoses in all cases, which means that although there was a decrease in the acid invertase activity, there were still enough hexoses. So apparently the acid invertase activity was not limiting, but another key enzyme comes into play, and that is the proton ATPase. The proton ATPase is located in the membrane of the phloem cells and also of the zinc cells, and it has a dual role. The first role is it activates acid invertase because this word acid means that the invertase has a low pH optimum and it needs low pH values in order to become active. We have seen that there was low activity, however, no limitation by this enzyme. So apparently the proton ATPase is limiting enzyme because it has a second role, and that is the activation of the hexose carriers in order to import the hexoses into the zinc cells. And this is actually the case. We have done in vitro experiments in order to measure the hydrolytic activity of the ATPase. And you can see that 2 days after pollination, there was a strong decrease of the hydrolytic activity relative to the control values. And this was more pronounced in Pioneer 3906 relative to Fabregas, where we also found this effect, but it was smaller. Now, the ATPase not only hydrolyzes ATP, it also pumps the protons, and we can measure this proton pumping in vitro. And what you can see is that Pioneer 3906 showed a decrease in proton pumping in contrast to Fabregas, which maintained proton pumping under the saline conditions. So what are the conclusions? First of all, ATP concentrations can be flexibly adjusted according to the plant's need, and a shortage of ATP does not occur under salt stress. Second, there is no salt limitation under salt stress unless plant tissue is severely damaged by iron toxicity in phase 2. Now, this may happen if there is strong iron toxicity. Of course, necrosis can lead to a dying of the leaves, but this is not relevant to our considerations because we would try to avoid this situation. Plant growth is limited by zinc activity in vegetative and generative growth phases. And finally, the results show very clearly that energy-saving traits do not deserve priority in breeding programs, but instead we should concentrate on the phases 1 and 2. That means we have to improve the osmotic adjustment. This is one point. And second, we have to improve ion exclusion and ion compartmentation in order to avoid ion toxicity. I thank you for your attention.
Thank you very much, Professor Schubert, for introducing the energy as a limiting factor to the development of plants. And you got, as I could watch on the window for questions, you got some questions that will be grouped to be answered at the end of the webinar for saving time. Thank you very much.
Thank you.
So next speaker will be Dr. Simbeswar Pratap Singh from Central Agricultural University, India, that will present us some case studies on crop nutrition management in salt-affected soils. So, Dr. Simbeswar Pratap Singh, the floor is yours.
Sven,
Yeah,
you
okay,
cut
sir.
the stop sharing, Sven.
Professor Schubert,
On the top. Okay, thank you.
Okay,
Is it visible?
we see your screen and please check your voice. That
Yeah.
Could you speak so we check that?
It's okay.
Yeah, yes, we hear
Yeah,
you.
yeah.
Thank you.
Okay.
Please go ahead, please.
Hello to everyone. And first of all, thanks to organizing committee to give me an opportunity to share about the findings of the work that is carried out by me in India. My title is Case Studies on Crop Nutrition Management in Salt-Affected Soils with special reference to India. I will discuss my presentation in following subheadings: background and what are the different interventions used to improve the salt-affected soils so that the nutrient availability could be enhanced, and conclusion and finally way ahead. In ancient period, based on the fertility of the soil, the soils are divided into two groups, urbara and anurbara. While they called salt-affected soil as usra, and these usra or salt-affected soils are named by different names in different parts of the India as well as other.
Shivishwar, sorry to interrupt you. We cannot see the change of the slides, and also we cannot see them in the full screen.
Now it is okay?
Yeah, thank you.
In India, the first systematic studies on problematic soil was initiated by Dr. J.W. Leather, and he also recommended to use gypsum along with the manures to reclaim the sodic soils. And considering the problem of the sodicity, salinity in India, Government of India, under the umbrella of Indian Council of Agriculture Research Institute, opened and a special institute known as Central Soil Salinity Research Institute in Karnal. Here is the status of the salt-affected soil in India. In India, about 6.7 million hectare lands are affected by different types of problems of salinity, sodicity, or coastal salinity. And if you see the distribution, here we can see that most of the Indian soils are sodic in nature, followed by salinity and coastal salinity. This is the special distribution of the salt-affected soils. Here we can see that in the states like Jammu, Kashmir, where the soil is acidic, or other hilly areas, the rest of the part of the India is affected by either salinity, sodicity at different extent. And here the status of the salt-affected soils in the state Bihar. As we already discussed that about in India, about 6.7 million hectares are salt-affected soils. And if you talk the state Bihar, there is about 4 lakh hectare area is salt-affected. And here we find a special type of problem in case of the salt-affected soils. Here, in spite of the excess exchangeable sodium percentage or salinity, there is also excess of the calcium carbonate in the soils. That's why here we call salt-affected soil as calcareous sodic soils or calcareous saline sodic soils. Likewise, it means here calcium is already available, but it is available in the form of calcium carbonate. And the solubility of the calcium carbonate is very less. And due to indiscriminate use of the fertilizers as well as the choking of the drainage system, the soils are becoming slowly converting into the sodic soils. And thus, owing to the less or non-availability of the pyrite, as we know that pyrite FeS2 is the best source to reclaim the calcareous sodic soils in Bihar. But due to less or non-availability of this source, can we go for an alternative source for the calcium, that is the gypsum? As we know that if we apply gypsum, the solubility of gypsum is more than the calcium carbonate, and it will ionize into calcium ion and sulfate ion, and this calcium ion replaces sodium from the exchange side. Likewise, if we add the pyrite, then it will react to form sulfuric acid, and sulfuric acid solubilize the calcium carbonate that is already available in calcareous sodic or calcareous saline sodic soils. So no doubt pyrite is a good source, but in spite of its less or non-availability, when we go for the gypsum, considering this in mind, we conducted this trial. And we also use different types of organic manures like the sulfonated press mud, that is the byproduct of the sugarcane industries. Likewise, we also applied the green manure in the through thatch. And here we can see that we selected about 49 farmers and about total 10 hectare area. And here we can see that for 2 hectare area, we selected 22 farmers. Likewise, in the bottom here, we can see that for 2 hectare area, we have selected only one area. But the basic idea behind this is that the land holding size of the farmers is different, and that's why their management practices are also different. And due to the different management practices, the management as well as the availability of the nutrient will differ. So considering these in mind, we collected the soil samples to characterize the different sites. And we put different type of the treatments like recommended dose of fertilizers based on the crop requirement, along with 50% of the gypsum requirement, along with recommended dose of fertilizers. In another treatment, sulfonated press mud at the rate of 10 ton per hectare. Sulfonated press mud is a byproduct of the sugarcane industries, and it contains calcium sulfate as well as the high content of the organic. We again added sulfonated press mud along with gypsum, dhecha along with gypsum, likewise. And in last treatment, we added all the nutrients. And we, as per the recommendation of the Central Soil Salinity Research Institute, Karnal, we started our management practices from the March. During March, we done labeling of the field. Labeling of the field is very much required and essential one, so that the gypsum could be uniformly distributed in the field. Again, the purpose of selecting this mud to start the management of the sodic soil is that we need extra water, and that extra water we will find through the rainfall. And here we have shown the step by step application of the gypsum in the field, and after that we mix the gypsum in the field, followed by the application of the irrigation water to release the sodium sulfate from the field. After that, we have shown the dhecha as a green manure, and after about 21 days, we have incorporated this green manure in the field. After that, there is transplanting. Here we transplanted the salt-tolerant rice variety, that is Usar Dhanti. And here we can see the status of the crop like rice, wheat in the field. Here is the yield of the rice. And here we can see that if we apply gypsum and sulfonated press mud, here we find the significant increase in yield in case of the sulfonated press mud. The reason behind, in comparison to the gypsum report, the reason behind this is that if we applied sulfonated press mud, it contains calcium sulfate, and the solubility of calcium sulfate is more than the calcium carbonate. That's why it produces the calcium. And the carbonic acid produced by the application of SPM will also solubilize the calcium carbonate that is available in the sodic soil. That's why here we can see that the efficiency of the organic matter is much more than the chemical amendment. But if we apply the gypsum, that is chemical amendment, along with the sulfonated press mud and dhecha, we find better results in comparison to the other soil. Likewise, here similar here is the yield of the wheat. almost similar trend was recorded. And here we can see that the extent of increase in the rice and wheat grain yield. Here we can see that there is drastic increase in the rice and wheat grain yield in comparison to the treatment where we applied only recommended dose of the fertilizer based on the crop. No doubt, as we know that in calcareous soils, the pH is very high, as well as the organic carbon content is low. And if we improve the soil by applying the organic matter or gypsum, then no doubt the availability of the nutrient will improve, and also there will be improvement in the pH. Here we can see the changes in the different soil parameters in the soils that was taken after the harvest of the crop, that is after third year. Not we have also taken soil samples from the surrounding areas of the experimental sites, and we have collected about 500 soil samples to see the variability in the problem of the salt affected soil in the surrounding area of the experimental soil. And here we can see that there is much more deficiency in organic carbon, as well as the pH of the soil is very high, and also the availability of the nutrient is low. The reason behind this is that due to high pH, the availability of the nutrients decreases. Thus, if we increase the two important key factors, that is pH and organic carbon, in the sodic soils, we can improve the nutrient status, available nutrient status in the soils, and that will help in enhancing the nutrient uptake by the crops. And another thing is that it is very important to aware the farmers. For that, we have also conducted different type of field day, etc., so that farmers could easily know what kind of practices in which fashion we should follow, so that farmers could get better yield. My last slide. Here is the conclusion. We found that the application of organic matter or organic amendment efficiency is much more high than the chemical amendment. The reason behind this is that there is already available calcium carbonate, and if we solubilize that calcium carbonate, then no doubt we could improve these soils. And here we found that if we apply organic as well as chemical amendment in integrated fashion, then the performance of the crop as well as the improvement in soil properties as well as the availability of the nutrient enhance. And the way forward, it is very important to aware the farmers, because the problem from place to place differs. So it is very important to aware the farmers. And another thing is that the farm holding side of farmers is different. So the timely availability of the inputs for the farmers, like gypsum, salt tolerant varieties of the crop, etc., is very important. Another very important aspect to improve the availability of nutrient is the site-specific nutrient. We should apply the nutrient based on the status of the soil. And another thing is that we should also think about the development of nutrient decision tools. There are different type of tools is already available, like nutrient manager, nutrient expert. So we can also think about the nutrient decision support tools, so that the farmers could use these tools to manage their salt affected soils. Thank you all.
Thank you, Dr. Singh, for your presentation of study cases on crop nutrition. And now next speaker is Dr. Nadia Bathihina, that will develop the topic of adverse effect on sodium and chloride on plants and strategies leveraging on saline soil heterogeneity to mitigate them. So Dr. Bathihina, the floor is yours.
Can you see my talk?
Yes.
Okay, perfect. So first of all, hi to everyone, and I would like to thank the organizers for giving me the opportunity to be part of this very nice and very important webinar. And so I will be talking about, as a quick outline of the talk, I'll be talking about why sodium and chloride are toxic in plants, and then I'll discuss whether they are equally toxic. This was also a point that was mentioned earlier as well. And then I'll talk about the potassium, okay? Potassium, how maintaining adequate concentration of potassium in shoots is very important for long-term salt tolerance. And then I'll move on to management options. And here I will focus on the heterogeneity of saline soil, how recognizing this and managing the heterogeneity can help us improve crop nutrition in saline soils. So I would like to start with this definition of what is a nutrient. There's beneficial and essential nutrients. This because, so this is a personal opinion, but sodium and chloride have, we mainly see the bad aspect of sodium and chlorides. And also when thinking about breeding, we often think about their exclusion. I come from a halophyte perspective, so sodium and chloride are not always bad. Actually, at low concentration, they're considered as beneficial nutrients. Of course, the issue in saline soil is that there's so much of it in the soils that in time, plants take up too much of that nutrient. And here is the optimal concentration for a nutrient. But with sodium and chloride in saline soil, you often, like the issue is that you quickly go above these requirements and you go over this critical threshold. And then you go into the toxicity range where you have toxicity when there's too much of sodium or chloride, and that then leads to toxicity symptom and growth reduction. But of course, something that is very important always to remember, the tissue tolerance, so the ability of plants to how much sodium and chloride they can have in the tissue will change quite a lot, depending on the plant species, and even within the same species, within genotypes and population, that will change quite a lot. So for instance, if you consider rice, which is quite salt sensitive, 100 millimolar sodium chloride would be too much for it, and that will tend to lead to plant death. Whereas when you talk about a halophyte, mostly the dicotyledonous halophytes, 100 millimolar is the optimal concentration, okay? The plant will grow even better than under controlled conditions without salt. So because halophyte can manage this, okay? They go and compartmentalize the excess of sodium, they put it where it's safe for the plant cells. So this is very important. So the threshold will vary quite a lot depending on the species, and that will affect the overall salt tolerance in the long term. So from a physiological point of view, so why are sodium and chloride toxic? So here you can see a picture where you see the visual symptoms of the sodium and the salinity toxicities. So you have leaf rolling, and then you have leaf injuries. So you'll have discoloration, chlorosis, and then that will lead to necrosis, which generally develops along the margin of the leaf tip, depending whether it's sodium and chloride. And then if the stress progresses, that will then lead to the whole leaf dying. But from a physiological, so within the plant cell, why are they toxic? First of all, too much sodium and chloride, as it happens as well. So there's an osmotic component, of course, when they are in the soil. But once they go in the plant tissues as well, for instance, in rice, we know that rice put a lot of that sodium in the apoplast, so in the extracellular compartment. What this does, it draws up all the water from the cell. So the cell goes towards, dehydrates, okay? And that leads to several issues, which is it affects the membrane integrity, and then it will make it much more difficult for the cell to function properly. Again, another effect, this is in part due to too much sodium. We know more about sodium than chloride, but too much of it will go and affect how the whole cell function, the whole metabolism will go down, will be affected. For enzyme, we know that enzyme, they require potassium. So if you have too much sodium, sodium will take up the place of the potassium, and that will affect the ability of the cell to function. For instance, with photosynthesis in the long term, if you have toxicity symptoms, that will affect the enzymes, okay? They are involved in photosynthesis, and then plant cannot photosynthesize anymore. But as well, another issue associated with too much sodium and chloride in the tissue is the oxidative stress. Okay, you have this production of these reactive oxygen species, which are toxic. So in a healthy cell, there is a balance between production and detoxification, whereas when the cell is stressed, then you will produce too much of it. And this excessive production of reactive oxygen species will go and attack the biological molecule in the cell, again, affect the functioning of the cell, and that with time can lead to cell death. There is a fourth aspect, but I will talk about that later. One little window that I would like to open, as was mentioned earlier as well, sodium and chloride, they're not always equally damaging, okay? There are specific, species-specific, some of them are more tolerant to sodium, more sensitive to sodium, and some of them are more sensitive to chloride. And really, the experiments that have shown this difference is the experiments where they've used a combination of salt. So they have the same osmotic components, but then they have either a sodium-dominant soil or chloride-dominant soil, and then they compare the growth compared to plant grown with sodium chloride. So here, for instance, you have experiments that were done with fava bean. You have two genotypes, a salt-tolerant, a salt-sensitive one, treated either with sodium chloride or with sodium-dominant and chloride-dominant, and with an EC at field capacity of 9 decisiemens per meters. Here you can see that the growth compared to control conditions, so here you'll have the growth under control condition, and this is the growth with sodium chloride soil. So sodium NaCl was the most damaging one, of course, but then when you go and look at the sodium and the chloride dominant soil, you can clearly see that the chloride is the one that is more damaging for the plant. So really for fava bean, you say, yes, there are some additive effect of sodium and chloride, but plants are really more sensitive. Fava bean is more sensitive to chloride, and that will be the case for citrus as well. When you move on to chickpeas, on the other hand, the history is completely different there, okay? So for chickpeas, they did the same experiments in hydroponics, and here what they saw was that this is the growth in sodium chloride, use 60 millimolar sodium chloride. As you can see, this chickpea, this genotype is very sensitive and really does not grow with salt, sodium chloride. When they used a sodium dominant salt, you can see that the growth is really the same as with sodium and chloride. On the other hand, when they had chloride salt, completely different, okay? In that case, here you can see that growth was not affected by the chloride salt, even if the osmotic component was the same, okay? So the growth with chloride salt was the same as the control. So in this case, it's clear the salt sensitivity in chickpea is exclusively due to sodium toxicity. So this is something that is really important to consider when we talk about the adverse effects of sodium and chloride in saline soils. Moving on, I'll talk about the last part, which is very important, which is the nutrient deficiencies in saline soil. And as it was mentioned, because there's competition for the uptake, of course, the chloride will compete with nitrate and phosphates. And whereas on the other hand, sodium has an antagonistic relationship with potassium, okay? So there is this issue that not only in saline soil, it's very difficult to take up the potassium because, of course, the transporters, they have it's hard, you know, that suddenly the concentration of sodium goes up in the soil, so it becomes very difficult for the transporter are not selective enough, okay, and they will pick up the sodium instead. But not only that, but the issue with salt stress is that it affects, okay, the membrane integrity, and not only is hard to pick up the potassium, but it's hard to keep it in the cell. So in plants that have been stressed, potassium starts leaking out of the cells. And this is a major issue for plants under saline condition because potassium is an essential macronutrient. Without it, the plant cannot function properly. So there are a lot of studies now showing that, yes, sodium and chloride is an issue, but as well a very important issue in saline soil is understanding to be able to maintain optimal potassium concentration in the plant tissues. And this is really clearly shown by a study that was done with the salt-sensitive spinach and the salt-tolerant quinoa. And as you can see here, so plants were grown with 500 millimolar sodium chloride, and as to be expected, so quinoa, the growth still went down with salinity, but you only had a 50% reduction in quinoa, whereas with with the spinach, the reduction went up to 80% compared to control conditions without salt. When you then, when you go and look at the sodium in the plant tissues, here you can see that really there's not much difference in terms of the sodium accumulation. So how much sodium goes in the plant tissue. So this is really, of course, but it's not the main factor leading to this reduced growth under highly saline condition. But then when you look at potassium, here the very nice difference comes, okay? So quinoa is very special. Quinoa, this is what I've seen in a lot of in my experiments as well. Quinoa, the more salt you put, the more potassium will pick up, okay? So here you can see these very nice differences. So in spinach with salt, you have a complete, so the plants lose up all that potassium, okay? The potassium concentration go almost close to zero, whereas in quinoa, as the salinity increase the potassium concentrations in the shoots. So overall, my take-home message here is that ion homeostasis is very important for the long-term salt tolerance. Okay, these plants have to perform for a very long time, they're always exposed to these salinities. So controlling, okay, how to take up the potassium and how to keep it in the cell. And at the same time, you also have to control how much sodium and chloride you're taking it up and where you're taking it up, okay? So this is critical for the long-term salt tolerance. Moving on, I'll go into the management options. So here I would like to draw the attention to one big feature of saline soils that is often overlooked, but saline soil are highly heterogeneous in time and in space. And here you can see an example of a field in the US, almond orchard, that is irrigated. And here you can see really this salt accumulation, okay, along the edges of the wetted zone. And here, on the other hand, is a map in agricultural soil in China, where in different times, okay? And here you can see salinity changes a lot in time and space. And the changes are quite substantial. You go from a few decisiements to, it can even rise above 50, the seawater level, and even more. And here, another thing that is very important to take in consideration is the issue of, we need to measure the salinity in terms of the salinity of the soil solution, okay? Because if your water goes down, okay, then the salinity will increase, okay? Measuring the ACO 1 to 5 EC, yes, but it does not give you an indication of the salinity that the plant are seeing. So that will increase even further the variation in salinity, because of course, water content changes at time and nutrients and salt as well. So I asked myself, okay, that was a question that was in the 1980s, Richard asked the question, because of these heterogeneities, also in terms of breeding, what should we focus on? Should we focus on the low salt areas or the high salt areas in terms of yield? And then I did these split root experiments where I went on and said, okay, let's see how the plant integrates all these different salinities that you have in the roots. And because I was working with the halophytes, I'm just going to show you an experiment where I really push the system to the extreme. And I went and look what happens if you only have 10 millimolar, which is a low salinity, in one root half. And in the high salt side, I went and put 3 times the seawater, so 1,500 millimolar of sodium chloride. And as you can see from this image, okay, it's very clear that under heterogeneous condition, okay, in this halophyte, the performance is exactly the same as the one in the control plant, okay? Whereas plants with uniform salinities, because it was very high, okay, this is a very tolerant halophyte, and so 3 times seawater was quite challenging for it. But what really came out from this study is that the plant with this, in this scenario, take up all their water from the low salt areas, okay? And this is also reflected in the sodium concentration in the shoot tissues. They had the concentration were very similar to those of control plants, and they were more than half those measured in the high salinity, the uniform salinity treatment. So really what came out from this study was that plants, that is where the water is coming from, that is very important. And in this case scenario, they took up the water from the least saline areas. Recent studies, however, have clearly shown that the situation is a bit more complex, okay, than I initially thought. And here they said, okay, we know that in saline soils, not only salt is heterogeneously distributed, but also all resources, so that means water and nutrients, okay? Because nutrients, especially the mobile ones, will be distributed in a heterogeneous manner, and that will change in time and space. So they did these experiments where they said, okay, so what happens if I move where the nutrients are, okay? They still have the split roots, have a freshwater side and a high salt side. And here they say, okay, let's put the nutrients in the freshwater side. And as you can see here, over the 9 days of the experiment, at the end, plants were taking up their water mainly from the freshwater, 100% from the freshwater side, and almost no water was taken from the high salt side, which is very similar to what I observed in my experiments. And that's what it means. Because all the water comes from the freshwater, you'll have almost no sodium and chloride in the shoot tissues. However, in this case, this is a glycophyte, it's a tomato. When you have nutrients on both sides, okay, the amount of water coming from the freshwater side will go down to a 70%, and the water coming up from the high salt side will increase to 30%. So plants not deficient yet in nutrients, but they start having more sodium and chloride in their shoot, which can become a problem further on, okay, as the time passes. But then they ask themselves, okay, what would happen if the nutrients are in the high salt side, okay, because that can happen. And here the result was very fascinating. So what they found was that, as you can see here, if for instance, potassium is not in the freshwater side, but is in the high salt side, almost all the water comes from the high salt side, which really means that for the plants, the nutrient is the most important, okay? They will take up the nutrient from where it is, even if it's in the high salt side, whereas water uptake from the low salt side, the non-saline side went down, okay? So that really raises a lot of questions. So is there a priority in terms of like, okay, is the salinity the issue, or also nutrients? We have to consider this as well. And to make it more complicated, they've seen that is nutrient specific, this response. Okay, when they did the same with nitrate, they found that you have a 50-50, okay? The amount of water coming from the high salt side decreased. So, but these have some serious implications, and we still do not know about it, okay? This is a very open areas, and we have a lot that we don't know in terms also of breeding, okay? What do we need to look when we look for breeding? Is there a scale? Is there which one is the more important signal? Is the nutrient? Is the salt? Okay, there's a lot to be unpacked here. But this, while it makes the management of saline soil more complicated, has also some really nice, it can facilitate, okay, the management of the saline soil. First of all, I think it's very important that we start recognizing this heterogeneity, okay? It needs to be recognized, and we need to recognize the different agronomical practices, for instance, mulching, green manure, that affects water retention. Will also, because they will change the amount of water, and that the salinity of the soil solution will go down, okay? It's important to start considering that different agronomical practices affect this heterogeneity, and we can use it, okay, depending on what we want to do, to modify plant performance in the field. And as well, the other aspect is that we know that irrigation, okay? Irrigation will create different zones of salinity, depending, so flood irrigation will create a low salt zone at the top, and whereas the salinity will increase as you go further down in the soil profile. Leaching fraction will affect that. But also here you can see the nice example where you see the different raised bed, the shape will affect where salt accumulates. And that goes, once we know that, then we need to really be careful when targeting the plants in the landscape, okay? Understanding this heterogeneity can then help us targeting the plants in the right place. So in this instance, for instance, if you place your plant right in the middle where you have that salt accumulation, you increase the chance that your plants will die. Whereas if you put them at the sides, that will enhance the chance that the plant takes up that low salt area, the water from the low salt areas, and the chance that your plant will perform better. But as well, so we've done some, for a review that we published in 2022, we did the modeling, hydrous modeling, where we say, okay, so we know that plants take up nutrients and water. Okay, we know the nutrient affect where the plant is taking up the water. So in terms of management of saline soil and fertile irrigation, so can we really use that to control? Okay, we manipulate where plants will take up the water. And we did this modeling with drip irrigation. And as you know, with drip irrigation, you have this typical salt accumulation patterns along. So you have the salt, you have low salinity close to the drip emitters. As you go away, there will be a flow of water and the salts that will tend to accumulate along the edges of the wetting zone. And if you can remember the picture I showed before in the orchard, the almond orchard, you see those circles, okay? This is the accumulation of salts along the edges of the wetting zone. So we know that happens, and the leaching fraction will affect these, where the salt accumulates. So we did the modeling and say, okay, let's think about fertirrigation. We know the nitrate is as mobile as chloride in soil, okay, and it will move similarly as chloride. So what would happen if I fertirrigate, I'll do 2 fertirrigation treatments. Here, I apply the same amount in both in A and B, but here I apply the nitrate, a small amount, but continuously, whereas here I'll apply larger amounts, but once every 10 days, okay? The amount of nitrate given is the same, but here is applied in a continuous manner, small amounts, whereas here is applied every 10 days, okay, so larger amounts. So we know that the more you put, the more you increase the chance of your nitrate leaching. And really what we found here with the modeling was that if you put low amounts more frequently, then you'll have an accumulation of dinitrate in the low salt zone, okay? And we know from the experiment that were done that the plant will really take up the water from the low salt area because you have low salinity and nutrients, okay? Almost all the water will come from there. But what happens when you put a larger amount every 10 days, then because you're putting a larger amount, the nitrate will travel down, further down, okay, in the profile, and will also will go also where you have the high salinity areas. And this becomes an issue because we know from the experiments that the plant will take up both the low salt water, but also the high salinity water, okay? Because nutrients are distributed in both everywhere in the profile. And that will mean that in time, plant will accumulate more sodium and chloride in the tissues, and then it will affect performance in the long term. So really, I think that this recognition of the heterogeneity of salinity in the field is critical, okay? It has some important breeding implication. They go, okay, there's a lot we don't know, okay? This is a field that hasn't been explored much, and unfortunately, a lot of breeding work is done under uniform condition, okay? You go then in the field, and the situation is completely different. So I think this is very important, but I think the nice thing is that we can also use, we know that we can also manage this heterogeneity, and that can help us improve the performance of the plants in the long term. Thank you very much for your attention. And I hope I've convinced you that heterogeneity is very important in saline soils.
Of course. Actually, there's variation in time and space. It is inherent to salt-affected soils and as well for irrigation. So thank you very much, Dr. Bathi-Hina. And I think that you will enjoy the field excursions during the meeting of Valencia next May. Now, according the program, I invite to Professor Muhammad Sahib to lead and dynamize a roundtable with the experts that have acted during this webinar, and as well, maybe Maria could address the questions that were collected in the window for questions to the correspondent speaker. So. Dr. Professor Saqib, I invite you to dynamize this roundtable now. The floor is yours.
Okay. Thank you very much, Professor Battle says. First of all, I'm very thankful to all the speakers for their very wonderful presentations today on this topic. I really enjoyed it, and I hope very much that all the participants, they enjoyed their presentations, in very informative presentations. And maybe I can request Maria, if there are some questions, we can pick up questions from the chat, and we can just discuss a few of them. And the remaining, maybe we can send to the speakers, and we can have their answer from the speakers, and then we can share in the group. So could you please state a few questions, then we can just discuss those questions.
Yeah, so I will collected some questions from the Q&A box. So first of the questions which I wanted to address to the whole panel to see your opinion on this question is that what do you think about the application? What is, let's say, better to apply foliar liquid micronutrients fertilizers on crop canopy rather than to apply it directly into the soil? So what do you think about this? What is more efficient way?
Okay, so anybody would like, Professor Schubert, would you like to respond to this question, or I should? So question is that whether nitrogen application as foliar application that is better under surfactant conditions or soil application that will be better.
Yeah, certainly we cannot generalize, and we would need a lot of time to discuss that in detail. But in general, of course, we have to distinguish between the total amount of micronutrients in the soil and the availability part of the micronutrients. And of course, there is, in many cases, a large discrepancy. So if the facilities are available, in many cases, I think it would be more efficient to apply the micronutrients on the leaves. This is not the case for the macronutrients because the amounts that we have to apply are much too high. But for the micronutrients, I would go for a leaf application in many cases. But of course, there are cases where this is not the case.
Okay, thank you very much. Thank you, Professor Schubert. I do agree with Professor Schubert that in case of micronutrients, of course, we can go preferably for foliar application, but for the macronutrients, maybe we cannot actually fulfill all the requirements. So maybe we can have to split it. Thank you very much, Professor Schubert. So maybe we can move to the next question.
Yeah, so next question is related to the organic matter. As soon as from the presentations, we can see that the organic matter management and application is very important for reducing the negative effect of salinity and increase the salinity, let's say, tolerance in crops. Then if the application of humic acid can it help reduce the impact of salt-affected soils on the let's say nutrient deficiency and nutrient let's say issues that we have on salt-affected soils if we apply humic acid to soils?
Okay, so Professor Pratap Singh, would you like to respond to this question?
Yeah. The major problem in sodic soil is the low content of the organic matter and high pH. And if we apply the organic matter or humic acid, then it will improve the pH as well as the organic matter content. And as we know that these two, pH as well as the organic matter content, is the key factor with respect to availability of the nutrients. So it will better, so it will respond more. And if we talk about the saline soil, then there is major problem of the neutral salts. So in that case, there is no need, we never apply any chemical amendment. We can go for the organic amendments. And we should also go for the agronomic management practices, as Dr. Nadia has already explained, where you have to plant where we have to put our seeds in which fashions. And also there should be a plan to remove the excess salts from the soils. So in saline soil, we should never add any chemical element, but we can add organic amendment, and we can also go for agronomic practices to improve the utility. Thank you.
Thank you, Professor Singh. Maria, would you like to share a few more questions, keeping in view the time, or?
Yeah, so let's, if we have time for 2 or 3 more questions. So there are some questions. So a small question to Nadia. Is there an ideal sodium to potassium ratio in plants? Or as long as sodium to potassium ratio below 1. Below 1 is an ideal condition. So I'm not familiar with this. Maybe it is quite familiar for you, such a question.
Yes, so here again, I'm really biased against this because I've worked so much with halophytes, okay? And halophytes accumulate a lot of sodium inside the tissues. And for the important is the concentration of potassium, okay? In my opinion, okay, the importance is how much potassium is in the tissues. And usually halophytes do tend to keep a nice sodium, they keep it constant in time. So I think it changes depending on the plant species. Okay, it's very plant species specific. Some will be able to tolerate lower potassium concentrations than others. So it's important to understand that. And to me, the critical is the concentration that you have and whether you put in the right place the sodium and the chloride. Okay, so if you, for instance, is all in the bladders, like for quinoa or saltbush, okay, then it's not toxic anymore. Okay, in the cytosol, you need to go how much is in the cytosol. Okay, how much potassium do I have there? And that really will change depending on the species. Usually with, so I think you need to do a research on which species you're planning to use and then seeing there how much potassium you have and whether you can keep it in the cells or instead of losing it all outside.
Okay. So I think that the last question, so as soon as we are a bit out of time, it is related to the beneficial microorganisms. So we understand that they can somehow increase the tolerance of plants, and they can they help increase energy usage and help through symbiosis to benefit both plant and animal and make nutrients more available to plants. What do you think about the beneficial microorganisms and so on?
I would like to ask Professor Schubert to please respond to this question. So what is the role of organisms, particularly under sulfur conditions? How can they be?
I didn't get the question completely, I think, but as far as I understood, there were two parts. One part was about the energy efficiency. In that case, as I showed in my presentation, this is not really relevant. Energy efficiency under saline conditions, in my mind, is not a factor that we should consider. It's not a limiting factor. The other thing is whether micronutrients, microorganisms can increase nutrient uptake. Yes, there are many examples that this is the case. However, it's very variable. So it's difficult to forecast whether there will be an efficient, a beneficial effect or whether it will not be. So it certainly depends on the situation. And I agree, there will be situations where microorganisms can promote the growth. Growth promoting bacteria is a big issue at the moment in the literature. And there are many examples that they can help, but I think it's difficult to forecast whether it will work or whether it will not work. Yeah.
Okay. So that was the last question, Maria, I think.
Yeah, so I suppose other questions I will share with the panel via email, and then I can share it with the participants after our webinar. Thank you.
Okay, so then I'd like to thank all the panelists for this short discussion, keeping in view the time we have to close this webinar. And all the questions will be responded by the panelists, and then we can share the answers with all the participants. So with this, I would like to ask Maria, would you like to have some remarks? And then maybe we can request Professor Betters for the closing comments.
Yeah, so actually, as soon as we are a bit out of time, I propose that we will ask Jorge to close the meeting. So if you are fine with this.
Yes, well, to not left any questions not answered, and as well for giving time to Maria to make the highlights of the meeting. I suggest that she will compile the questions and address to the particular speakers to be answered, and with these answers, this will be again emailed to the participants. And well, I want to thank Dr. Professor Sahib for his excellent organization and finding the specialists on the topic and joining them here in this webinar, and all attendants to the meeting that make that that encourage us to follow in with the series of webinars that until now has got a crowded participation. And before closing the webinar, I would like to make an announcement, and it is that we INSAS joining with an action cost devoted to the sustainable use of salt-affected areas with plants. We will have such event, let's say a symposium together in a coordinated way at Valencia last week of May, next May. So it is just one month from now to the celebration of this meeting. And I will share with you the link in the chat window to where you will find all information about this meeting. And I invite you all to participate in online or in person. If you come to Valencia, you will be welcome. And anyway, this has to be a hybrid event, so people in remote areas can attend and participate in discussions and learn about what lecturers present in this meeting. So thank you all of you, dear colleagues. I'm very glad of seeing that participation was from quite any longitude in the globe. And so let's meet soon, or in Valencia, that you will be welcome, or in next webinar that will be scheduled soon. Thank you all for your participation.
Thank you. Thank you.
Thank you.
Thank
Goodbye.
you. Bye. Thanks.
Thank you.
Thank you.
Maria, it is possible to meet in another room for 5 minutes? Yes, please.